Hello, I'm Dr. Olga Economidou, academic consultant, medical oncologist, and breast cancer translational research group leader. I work at the University of Edinburgh and the Western General Hospital in Edinburgh. A few years ago, the University of Edinburgh and my group had the privilege of being awarded the first grant by Make Seconds Count. The grant was used to fund a 3 years postdoc fellow position, and the project that was assigned to Fiona Semple, who was employed in this post, was to look at the underlying mechanisms of progression of a very aggressive breast cancer subtype, which is the triple-negative breast cancer, looking at the evolution of the cancer from the very early setting to the advanced metastatic setting and subsequently to death. We are currently in the position of analyzing our data with the help of the bioinformaticians at the University of Edinburgh, and we hope to present our data in the next couple of months in one of the international breast cancer meetings. We used very high-end modern techniques, and in the video that will follow, Fiona will be able to take you through to the project and the current results. We will always be internally grateful to Make Seconds Count for giving us the opportunity to run this project, and we will always be grateful, of course, to our patients that they have donated their tissue and biological material so as to be able to look at these changes and all these underlying mechanisms I just mentioned. Thank you very much.
I'm Dr. Fiona Semple. I'm a research scientist in Dr. Economidou's lab, and I'm going to describe some of the lab work I'm currently doing on genomic profiling of triple-negative breast cancer. Many breast cancers are driven by estrogen, progesterone, or HER2. So if you think of the tumour as a car, some are fueled by estrogen, some are fueled by progesterone, and some are fueled by HER2. So these cancers, we know, can be treated with drugs that specifically disrupt that supply of fuel. And this is how hormone therapy works. It stops estrogen, HER2, and progesterone driving the cancer. However, triple-negative breast cancer is not fueled by estrogen, HER2, or progesterone. The fuel that drives these cancers to grow is unknown. So triple-negative breast cancers have to be treated with a one-size-fits-all chemotherapy and immunotherapy approach. So there's a definite need to find more specific treatments for triple-negative breast cancer. 15 to 20% of breast cancers are triple-negative, and it's more common in younger women and not treatable with hormone therapy. It's also a cancer that spreads quickly and is more likely than other breast cancers to spread to other parts of the body. It's also more likely to recur after treatment, and most patients with TNBC are treated, as we said, with chemotherapy and immunotherapy. However, this blanket approach to treatment is potentially the reason for some of the poor outcomes with TNBC patients, as the treatments that suit one type of TNBC tumour may not be the best for another. And this is because every tumour is unique. So one way that we can explore the differences between TNBC tumours is to investigate them at the level of the genome. So the genome is the complete set of DNA in each cell in the body. It's packed into the nucleus of the cell in 23 pairs of chromosomes. And if we zoom into this chromosome, we can see the DNA molecule, which is shaped like a twisted ladder, with the rungs on the ladder being made up of these base pairs A, T, G, and C. And in each of our bodies, in each of the cells in our body, we have more than 3 billion or so base pairs. And we represent the DNA sequence using these 4 letters A, G, T, and C. And this is important because cancer can be described as a disease of the genome. So each of the cells in the body divide to make new cells, all of which contain a new copy of the genome. However, during this process, sometimes changes occur, such as in this cell here. And these changes are known as mutations. But usually, the mutations are detected and removed by either repair or cell death. So this is what happens in normal cells. However, in tumour cells, when mutations occur, they're not detected, so the tumour can continue to grow and accumulate more and more mutations. And often these mutations drive the progression of the cancer, contributing to all the uncontrolled growth. So the genome of TNBC tumours is complex because there are different types of mutations. So tumours can contain single changes in the DNA, like the one shown here, where the normal DNA has an A and the tumour has a T in this position here. This is a mutation known as a single nucleotide variant. However, cancer cells can also accumulate larger changes to the DNA. These are called structural variants. And here, large chunks can be lost from the chromosome, such as when the chromosome breaks and we get a deletion here. Also, large areas of the chromosome can be copied and we get what is shown here, which is a duplication. And we already know that these structural variations, both deletions and duplications, are a very common finding in triple-negative breast cancer. So here in the lab at the IGC in Edinburgh, I'm investigating both types of mutation, the single nucleotide variants and the structural variants in TNBC tumours. To do this, we use advanced methods based on DNA sequencing, and there are lots of different assays to profile variants in the genome. However, our aim here is to investigate some affordable methods that can be employed at scale with large numbers of samples from all patients from the clinic. So because not every sequencing method is good for profiling all variants, I have two methods up and running in parallel. First of all, whole exome sequencing, which is good for the detection of the single nucleotide variants, the small mutations, and cut-seq, which is good for detection of structural variants, the larger mutations. And unfortunately, I don't have time to describe either method in a lot of detail, but the important thing to know is that by combining these two methods, we obtain a genome-wide assessment of both the small SNVs and the larger structural variations in the TNBC tumours. So just to give you an overview of what is involved, firstly, we receive the tumours in the lab and the DNA is removed from the tumour cells, and then the DNA is prepared in a multi-step process which then allows it to be read by sophisticated sequencing machines. And the output from these sequencing machines is many files containing billions of DNA sequence reads. So those reads are then compared to known normal copies of the human genome, and the altered DNA sequence is then identified. And this process where the data is interpreted is known as bioinformatics. So just to reiterate, what we're doing here is reading the genome of the cancer to find the mutations, both the small mutations and the large mutations. And we do this by taking the DNA from a patient's tumour sample and a patient's blood sample, and we compare them. This is done using genome sequencing to read the DNA, which is then interpreted using bioinformatics. We then find the differences between the patient's normal DNA and their tumour DNA, and these differences are the mutations. So identification of particular mutations in a patient's tumour is very important because we already know that specific mutations can inform on whether or not a tumour will respond to a therapy. So this can help direct therapies to patients rather than the one-size-fits-all approach of chemotherapy. So we're currently accumulating results from around 100 samples from TNBC patients using both whole exome sequencing and cut-seq. And at the moment I can share some results from our cut-seq approach. So here we have the results from one patient, and this shows data from the DNA across the whole genome. Each column on this plot represents a chromosome from chromosome 1 to chromosome 22, and each dot is a point along the genome. And this is the normal blood sample from this patient, and we can see that it is approximately a straight line, excuse me, which means that there's not much structural variation in the genome of the blood. So this is the baseline that we compare to the tumour. So in comparison, here in the tumour, from the same patient, we can see that there are many areas where parts of the genome have been duplicated. These are shown in red. So these are areas such as here on chromosome 1, on chromosome 4, 5, 8, 17, 18, lots of areas of duplication. There are also other areas where parts of the tumour genome have been deleted, and these are shown in blue in these chromosomes. So what we can see, just in general, is that there are many rearrangements in the patient's tumour DNA compared to their normal baseline DNA and the rest of the cells in their body. And using this sequencing technique, we can track these changes across the course of a patient's tumour. So we have here in Edinburgh a very unique set of samples collected from patients. So we have samples from the diagnostic biopsy, and this is usually from before any treatment has occurred. And then we have samples from surgery, tumours removed at surgery, which will be after pre-surgery chemotherapy. And for some patients, we also have biopsies from tumours that have recurred. So this allows us to investigate DNA changes throughout the course of the tumour. So in this patient, we have a blood sample, a tumour biopsy, a surgical sample, and a recurrence sample. So here we can look at the duplications and deletions throughout the treatment of this patient. So again, as before, with the normal blood sample taken from the cancer patient, we can see very little variation across the genome. In the diagnostic biopsy taken before the patient's treatment, we can see again extreme changes in the DNA structure across the genome, with specific increases on chromosome 1 here, chromosome 2, chromosome 8, chromosome 13, amongst others. And then if we look at the surgical sample, you can see that in this sample, compared to the biopsy sample, a number of changes have occurred. So for example, there is now a deletion on chromosome 1, which wasn't there in the biopsy sample. There's increased duplications on chromosome 1 compared to the biopsy sample. There's the disappearance of a deletion on chromosome 4 and chromosome 5, which are here but not here. And there is also the appearance of a deletion on chromosome 14 here, which isn't there in the tumour biopsy sample. So these differences in the surgical sample tumour genome may be as a result of the chemotherapy that the patient has had before surgery. But what this suggests is that the pre-surgery chemotherapy has altered the evolution of the tumour. So the tumour has now evolved to have different structural variants, and some of these changes might be associated with the different patient outcomes. Once we've sequenced multiple samples from hundreds of patients, these are the types of questions that we will be able to answer. So in the case of this patient, unfortunately, the patient's tumour has recurred, and interestingly, we can see that it shares some of the changes we see in the original tumour, such as duplications on chromosome 1, which are seen in the original tumour, chromosome 8 in the original tumour, chromosome 15 and 17 and 13, which can be seen in the original tumour and here. And these particular changes may give us clues as to why and when a tumour recurs, and that may suggest new ways to stop recurrence. So to summarise, by sequencing DNA from a tumour and comparing it with normal DNA from the patient's blood, we can identify variants in the tumour. And knowing which tumours contain which variants allows clinicians to use appropriate drugs to treat these patients. So at the moment, we can use what we learn from the genome of a tumour to target the drugs we already have to the right patient groups. And this will take us beyond the one-size-fits-all therapy and allow more specific treatment for each patient. So in terms of future, tumour sequencing studies allow us overall to have a better understanding of tumour biology, in addition to identifying potential biomarkers of response to chemotherapy. Sequencing may also suggest new genomic targets for treatment and may even suggest new options for drug development. And all of this will lead to accelerated clinical progress for triple-negative breast cancer. So finally, I'd just like to thank everybody involved in this work. Firstly, all of the patients who very generously donate tissue and blood samples. Devon and Colin, the bioinformaticians. Natalie Wilson, who's a tissue consenter and involved in sample collection. Alistair Ironside, who's a pathologist who's involved in the identification of the cancer cells. And the clinical research facility, where all the sequencing happens, and Make Seconds Count, and the BCI for funding this research. And thank you very much.